is Alex Pellegrini, and I'm going to explain the protein structure. First, the protein structure starts with the primary structure, which is a linear sequence of amino acids that form a polypeptide chain. This polypeptide chain is determined by the DNA of the gene that encodes the protein. And it kind of looks like this. This is also the simplest level of the protein structure. And it is formed with peptide bonds, also called amide bonds. And these bonds make up the entirety of the polypeptide chain, which is what I'm drawing here right now. Protein can be made up of a single or many polypeptide chains. It is made up of covalent bonds, which is a very strong bond, and it is formed between carboxylic acid and amino groups by the loss of a water molecule. It is very strong, stable in water at a neutral pH, and stronger than a typical C to N bond, carbon to nitrogen. So it kind of looks like this, and it has an amino group right here, an acidic carboxyl carboxyl group, an R group, and carbon and hydrogen. Well, next is the secondary structure, which is the linear sequence of amino acids, and this folds upon itself into folded structures that form within a polypeptide chain due to interactions between the atoms of the polypeptide chain, except for R group atoms. It can either fold into a beta pleated sheet or an alpha helix. Alpha helices, plural. The bonds that make this up are hydrogen bonds, and they form between one of the lone pairs of an oxygen atom and the hydrogen atom attached to a nitrogen atom. Hydrogen bonds are one of the strongest bonds, but weaker than covalent bonds, which are found in the polypeptide chains in the primary structure of the protein. So, the alpha helices kind of looks like this. I will do a quick sketch. And so on. And the pattern varies. It begins with nitrogen to carbon, a double bond to oxygen, a bond to hydrogen. Nitrogen also bonds to hydrogen. And then carbon, nitrogen to carbon to hydrogen to an R group, and the carbon bonds to an oxygen, and then There's also a bond between this hydrogen and this oxygen. And the beta pleated sheet kind of looks like this as it folds. And starts out with the pattern carbon to nitrogen to carbon to carbon to an R group to hydrogen to oxygen. These bond to carbon, Carbon has a double bond with oxygen. This carbon has a bond to hydrogen. This carbon has a bond to nitrogen, and this pattern continues between these two beta pleated sheets, which will fold together and bond as well in between. Next is the tertiary structure, which is the final overall three-dimensional structure of a single polypeptide chain. It occurs when certain attractions are present between the alpha helices and beta pleated sheets, which are these. So this is the alpha helices right here, the squigglies, and beta pleated sheets. It is a higher order of folding within a polypeptide chain and depends on distant group interactions. It has hydrogen bonds, ionic bonds, hydrophobic, and some covalent bonds called disulfide bonds. It also has dipole-dipole interactions, and London dispersion forces are also present. It's typically pretty weak, and it occurs between the groups of amino acids. So, again, alpha helices and beta pleated sheet. And finally, we have the quaternary structure. So this describes the bonding between multiple polypeptides, and it occurs when a protein consists of more than one amino acid chain. So basically, if this was one crazy amino acid chain, it would bond to this yellow other amino acid chain. And you can have more than two of them, but just for now, I will stick with two. The bonds between these can be hydrogen bonds, ionic bonds, sometimes covalent bonds, and van der Waals bonds. These are mostly weak bonds, and it occurs right here between R groups. And then altogether, you're going to have your protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. The end.